To start with, my name is Markus Loeffler, forget about all the titles. They are not relevant here. I do welcome you to Kunstkraftwerk, and this is now going to be quite a different talk. It's more a science talk, and I'm curious how this will work. I should perhaps first mention that I'm a hardcore natural scientist. I have a training in medicine. I have actually an MD degree, but never practiced. I have done a study in theoretical physics, but I declined any offers for a dissertation there, and instead I went to do something very different, which nowadays is called systems biology, systems medicine. It's about modeling biological processes. In a certain sense, this is abstraction from the real facts. But this didn't give me a living over 30 years ago. So in order to make a living, I switched to medical statistics. That gives a living. It's a lot of work to do, and that's my profession. What I want to tell you is a little bit, and by the way, you realize the difference in lectures. Science lectures are slide, 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 while the other ones we had this morning were more narratives. So this is, I'd like to discuss concepts of tissue organization, in particular of regenerative tissues. These are the ones who are renewing every day, and they need to have particular organizational principles. And I'd like to show you how one can try to understand these organizational principles by trying to model these tissues in the computer. And at the end, I'll like to give you a little bit of an idea on what I, as an art dilettante, could imagine as a little sort of project to go into an art performance. By the way, we have been doing a lot of these modelings for different regenerative tissues. This is just a list of what is all regenerating. Well, of course, it's the blood formation. It's the skin is renewing. It's the connective tissue is renewing. The liver is regenerating. The cartilage is slowly regenerating. The intestinal system, and that is the gut, is regenerating a lot. And of course, tumors are also misconducts of regenerations. I'll like to kick off with this little photo of a swarm formation that you can see here with some fish. They are organizing in a particular way to create large macroscopic pictures and structures. They resemble kind of organisms, but there is no organizing instruction behind it. There is no unique brain behind it. It's a collective phenomenon that leads to these organizations. And you have similar things not only with fish, but with birds. You have it with social insect states. There are very similar organizational principles. And now I ask the first question to you. What would be a simple set of rules to explain this phenomenon? I'll give you a few rules that one could think of. And this is, the phenomenon can be very well explained if just two rules apply. Each of the individual organisms in this swarm obeys two rules. Number one is move slowly but steadily. And number 2 is stay close to a fellow. If the 2 things come together, then under particular circumstances, such patterns arise. And that's also already the basic idea for my suggestion to do a performance. When biologists try to understand such things, they are not thinking in terms of rules, because rules cannot be measured. If you want to examine such a thing, you will, to make it a little bit silly, you throw the fish into a mixer and you get out the gene expression of their brains, of their muscles, and then you want to understand how they interact with one another, and you'll find a lot of correlation structures, but nothing in the biological system tells you what are rules. Why is that? That is because rules cannot be observed. They are cognitive concepts. They are abstractions. They are something that originates in our mind when we try to understand a process, and this is a composite. Nowhere in nature is there a rule written down, except on when Moses found the 10 rules on the mountain. So it's a little bit playing with these things, and I want to illustrate you how a set of rules can be promoted to explain a large set of observations, and in this case it's about tissue organization in the intestinal gut. When we're putting together such rules, we don't want to do, there is a sort of parsimony principle. Keep things simple as long as you can. Try to make things understood from what is called Occam's razor. Do only add those ingredients of rules that are really required and leave everything else out. So it's a reductionist approach. I'll introduce you a little bit to the biology of this system. So if you open the small intestine, it looks a little bit like this. There are these little finger-like protrusions into the lumen of the gut. These things are called the villi, these fingers. And between these villi, there are little buckets in the wall of the gut, and they are called crypts, very much like the crypta in a church, because it's somewhere down where you have to go, hidden structures. And these crypts, you see here, this is the outer part of a gut, this is the inner side here. You have these fingers, villi, and then you have these things called the crypts. And what you can see here in this picture is there is a sequence of cells. It's only one layer of cells. They are covering this sort of connective tissue structure. And actually our meal at the moment is coming along here. It's taken up here, and then it's going to the bloodstream, and that's how you feel now pretty comfortable after our lunch. Now, this tissue here is constantly moving upwards and is shed off. In human, this is happening, total replacement of the entire one-layer tissue each 5 days. Total replacement. And where do the cells come from? Well, they are coming from these buckets, these crypts, and there is heavy cell proliferation going on. And there are a couple of different cell types one has to consider. There are, you see it from the staining, there are particular cells, they are called Paneth cells. Then there are a couple of cells I'll show you in a minute. Here you see them. They produce mucus, and as they are producing it, they having this sort of goblet type of character. That's why they are called goblet cells. In Deutsch heißt das Becherzell. It comes from the Weinglas. weil die wie ein Weinglas aussehen. In English, the goblet is the calyx for wine drinking, and they have a little bit that shape. So old descriptions, old names. Now, here are other pictures of it. There is staining properties that you can generate. Dutch researchers have found there is a staining which is very likely staining the intestinal stem cells. Those cells which are sitting there more or less for a very long time period and regenerating all this tissue. If we are looking on the fingers, on the villi, then there are single cells spread out. These are the goblet cells. You see them now from a planar projection. And it is interesting to see that they are isolated. How can they be isolated? If the tissue is coming up from the bottom, what makes them single? How is this organized? So there is a constant streaming of the tissue, and there is a constant differentiation of at least 4 different cell types going on. Then there was another observation, which I owe to a colleague, disease colleague, Chris Potton from Manchester. There are particular animals where you can do a little harm to the genes, and these are then genes knocked out for staining properties. So these cells and all the progeny descendant from them don't pick up the stain which the others have. So you see a ribbon of cells moving up, these villi. They are all coming from one stem cell at the very bottom, and all the descendant cells, daughter cells of dividing cells, lack this property. Now, it was observed, first of all, that these ribbons, you see several, have a fluctuating size, and the crypts where they come from, they, in the initial phase, they share the white and the brown color type of cells. But after 100 days, they convert to either be fully filled with unstainable cells or with only stainable cells. So there is a what we call monoclonality conversion. There are other pictures of such ribbons showing that they are a little bit fluctuating in width, and these are just the data saying that it takes 100 or 150 days, and there are only monoclonal crypts, and there are no mixed chimeric crypts, if you like. So they are washed out. Also, this is a phenomenon we want to explain with the same type of rules, same set of rules. The first model I'd like to show you is a little older one, but it's very nice for illustration purposes. Actually, there is the idea that in such a crypt, we have a heavily proliferating zone, and a dividing proliferative cell generates 2 daughter cells, and they would also proliferate and continue with this. Once though they have crossed a certain border, they would switch the program, and then it would be decided they would go for maturation, stop proliferation, and prepare for the work of picking up and digesting nutrient. This is a very simple rule. So the rule means we need to set up a model. This model is a very simple one. We have a kind of water glass tube, a rigid one. On the surface of this, which is a closed cylinder, we display cells. The cells are polygonals, and then we need to find some rules on how they grow as the cells get larger and larger, and they split, and then they move away the other cells in their vicinity just by pushing them away. So with such rules in place, for example, we can, it should work actually, yeah. You have this, I'll repeat this, it's a short movie only. And here you see there is this threshold. Once they cross, they convert into green. All the other ones remain yellow. They are all proliferative, and they have all the same rights. So these are all the same fish, if you like. And the tissue streams, it goes up, it produces cells, and so on. Okay, if we now give a little color just to one cell here at the bottom, it has no particular other qualities except that this blue dot is now carried forward to all its descendants. You'll expect a sort of ribbon evolving, and we'll follow such a fluctuating ribbon of labeled cells. And you see wild fluctuations, and they're just occurring because of the local ordering of these cells, which are aligned. And in this case, for all the simulation time, it was mixed. It was, I'll repeat it once again, there were labeled and unlabeled cells here. In this case, I changed it. I made the environment a little bit bound. And this leads to the fact that the monoclonality conversion is much more rapid than if I have a flat sort of surface. So in this case, I'll repeat it. It is clear I have one here sitting here in the valley. This one is the lowest cell at all. It can easily shift all its descendants uphill and thereby conquers the clonal population. If the labeled cells would be sitting up here on the hill, there would be almost no chance that its descendants would climb down. And this is just the little local disadvantage of this swarming tissue of this cell to make the clone. So the simple prediction is the lowest cells in the crypt should be those with the best clonal formation ability. Repeat this once again, and you see, after a while, all are labeled. Now, you can restrict these things. For example, if putting such little buckets in. This, okay, this constrains the effect, and you have a localized clone formation. Now, this was a very simplified model. It was a rigid tube. It didn't consider lineage specification. There was only proliferating and maturing cells. Well, now we want to go further, and we make a more advanced model. And this model here is placed on a meshwork of elastic fibers, which we construct in the computer. So there can be tension and forces can be exerted. On this meshwork, we place single 3D spherical cells, as you see here on the left-hand side. And then we introduce now rules not only similar to the ones that you have seen regarding migration, but also rules considering lineage formation. So now there is the question, what singles out goblet cells, the yellow ones from the blue ones, and what makes the Paneth cells? Now, there is a lot of biology going into this consideration. I'm only roughly touching on this. It has to do a lot with biomechanics, with biophysics. So elasticity is coming along, forces are exerted, but also you need to have rules on the cell fates. But it's all about rules. And only the set of rules now gets a little bit more complicated. Biologists tell us that there are two particular signaling systems in this tissue. They are very active. They're having names for this. One signal system is called the Wind signal. This actually is basically creating a gradient up the crypt. And if this gradient of signal, if the signal in the environment is high, that means at the bottom, this is encouraging cell proliferation, and if it's going low, it's leading to shut off of the proliferation. So this is basically the threshold that we had a minute ago. The other system is called the notch signaling system, and this is a local, immediate next neighborhood interaction, and basically leads to the fact that if one cell says, I'm producing notch, communicates it to the immediate neighbor cells, it blocks their notch formation and inhibits this. So this is a lateral inhibition process which controls that one cell keeps its particular feature and all the surrounding cells don't do the same thing. So this is the process of individualizing the cell differentiation. So they are simple, again, from this biological background, we distilled, if you like, 2 rules, and the rules are, wait a minute. The rule is this notch rule, it's the next neighborhood rule, expressing cells suppress lateral neighbor cells to do the same thing. And in this context, the effects of this inhibition are different whether you are high up in the crypt and then cells are creating Panis characters or not. And if you're low in the crypt, meaning high wind signal around, then it's the decision of whether you are stem cell or Panis cell. And the wind signal I already explained. This leads finally to this simple table. At the bottom, you have 4 different cell types: the red one, the blue one, the green one, the yellow one. And the decision which one is taking which character depends only on which of these rules apply, the next neighborhood rule or the signaling rule. When this is made to work, then you get such simulations, and this complies very well, which I'm not going into now, with biological scenario. Here now we have the clonality conversation with a pink ribbon coming up, and you see this is now filling the same crypt, it's just split open and displayed. So the clonality conversion works well. We can also simulate genetic manipulations. Let's say if the rules would be killed away and destroyed, what would happen in this case? The crypt would be fully populated with mucus-producing goblet cells, and actually such things are observed. There's a whole set of experiments around to which we can make this model fit simply by deleting the rules, overexpressing the rules, making them more rigorous, or so, fine-tuning of this. I will now, so you get a flavor of this, how this works, and I will jump to my suggestion, and I briefly talked over this a couple of months ago with Gustav Hellberg, but we didn't dwell on it any further. But I thought, and now this is my dilettante art contribution, to make swarm formation maybe go into a performance. Now let's imagine, that's my sort of simple introductory remark, but I'm sure you are all having much more creative ideas about this. I imagine a dark room in which we have a certain number of persons. And they are moving around slowly. And they wear hats with some illuminated sculptures on their top. Something like this, what you see in the Basel Morgenstreich in carnival season in the night. People are walking around with little lamps on their hats. Something like this, or like this. Yeah? And you have a rather dark room, and then you have a video recording from the top. So you see how all these little dark light things move around. Now each person will be given a set of instruction cards, which are pulled out one after another, let's say, upon a whistle blow. And now the first simple scene would be people are in the room, and then the first instruction comes, and this is only card A, and says, move slowly but steadily as you like. What is going to happen? Well, a random walk. So there will be a random walk, and then you can start to activate card B. And card B would simply say, search to stay close to a companion, but let's say not too close, not heart to heart, but at a decent distance. What is going to happen? What would you expect to happen? Well, this is an experiment we can do. And this is an experiment on which we can play, really. We can have many people, not so many people. We can influence the velocity of the walking. We can influence the distance between the persons, the proximity. But if we are doing it right, we'll get at least a chain, and sometimes the head of the chain is meeting the tail of the chain, and we'll have a circle. Actually, we did this experiment with my institute a couple of years ago with about 100 people participating, and it took about 5 minutes to get this circle. It really happened. And I told everybody, don't look for a search. I didn't even tell them what I was looking for in terms of observation I wanted. But there was a circle then. Now, this is something to experiment on, very clearly. There are many parameters on which you can fiddle around, and then you can go a little further. Now, let's say such a circle has formulated, and it's working. It's going round and round and round and round, and then you can introduce another intervention. Now you activate a third rule, which only a few persons have. Only a few. And this would, for instance, say, leave the swarm and move towards the exit. What is going to happen? Well, something to experiment on. The question is, under which conditions will the circulating swarm break down? Is one person exiting enough? How many? In which kind of orchestration would they have to leave to break it apart? This, in a socio-ecological experiment, would mean how many opinion leaders do you need to make a long chain of people follow you? Just think about this. So another set of experiments, and of course, there are many more such possibilities you can think of, and I imagine this would be a possibility to do own experiments on with a set of artists. I think it is fun. It takes an hour or two. And then I think to make from this, to go on and to carry it forward, to make it to a little bit more fascinating introduction into how patterns form, in how patterns break down, into what are we call it in science, the phase transitions that are occurring, the bifurcations. But this, I think, is a part of science, and the other part is with art. I hope I have given you a little bit of inspiration, and I'm hoping for many more for me. Thank you.